Good morning, everybody. I'm Jennifer Van Gaskin. I'm a principal investigator and lead the Ocular Fibrosis Research Unit here at Sarah, and also a consultant ophthalmologist at the Eye and Ear Hospital specialising in glaucoma. My research is on developing new ways we can prevent diseases in conditions that cause scarring in the eye. I would like to acknowledge and pay respect to the traditional custodians of this land. I acknowledge that the land that we meet and work upon is the traditional lands of the Wurundjeri people, and pay my deep respects to the Woi Wurrung elders past, present and emerging, and to all elders of the Kulin Nation. Indigenous Australians over 40 still have 3 times the rate of vision loss than non-Indigenous Australians. As we approach International Women's Day, I pay tribute to the many First Nations women who are working tirelessly to create better outcomes in eye health for their communities. Some orientation for those of you who cannot see me. I'm standing at a lectern at the front of the room with a large screen behind me, which currently has a holding slide with an image of me and my title. The speakers today will explain any images on the screen for those who may be having trouble seeing the slides. It is a great honour to be here today for this event in the lead up to International Women's Day tomorrow. I'd like to welcome those of you who are here in person and online, in particular the Sarah Board Directors, Professor Andrew Cuthbertson AO, who is also Deputy Chancellor of the University of Melbourne, special guests Jackie Marie and George Samus of GJK Facility Services, who are here today to unveil a key piece of equipment which will benefit research to improve women's eye health. Our amazing Sarah staff and students, and Dr. Anna Wang, who will today be giving her first research presentation as Sarah's inaugural Equity Fellow for Excellence in Vision Research. A special acknowledgement to the many philanthropic supporters who make our work possible, many of whom are women, and a personal shout out to some of my philanthropic organisations that have supported my research, including Australian Vision Research, the Cass Foundation, Sarah's Innovation Fund and the Dorothy Adele Edels Charitable Trust. The theme of tomorrow's International Women's Day is Count Her In, Invest in Women, Accelerate Progress. I'm very excited to be here today as part of an event which is celebrating a major investment in one of our women scientists here at Sarah, and also to acknowledge a research project that focuses strongly on women's eye health and a generous donation that is making that research possible. On International Women's Day, we can reflect on the progress that has been made by women in science, and also the huge amount of work that needs to be done to achieve equity, to achieve true equity. Earlier on in my career, one of the greatest challenges I faced as a young female researcher was to be taken seriously. I knew this because my research supervisor asked me what I was going to wear to a presentation when I was very early on in my career. And that wasn't said in a way that was sleazy or inappropriate. He was trying to help me, but he said, don't wear a dress because you don't want to be seen as a little girl. I don't like to think that it was that long ago when that happened, but I do think times have changed. But it is only by increasing the diversity of people standing in front of at the podium, the diversity of people sitting in the audience, and the diversity of people who get to dictate what presenters wear that we can truly make a difference for young women today. When I was a medical student in Auckland, I almost ruled out a career in ophthalmology because I was told that it was impossible to get in. And it's taken a lot of persistence, resilience and hard work to become an ophthalmologist, but also to juggle my clinical commitments with raising a young family and a career in research. But progress for women and other underrepresented groups in ophthalmology and in science shouldn't just be about personal effort. It is also about making sure that organisations address the barriers that unfairly impede progress for certain groups of people. And that is why initiatives such as Sarah's Gender Action Plan and Equity Fellowship are so important. Thank you to everyone for joining us today. I'd now like to introduce Sarah's Managing Director, Professor Keith Martin.
Well, thanks, Jen, and I'd also like to acknowledge the traditional owners of the land where we meet now, the Wurundjeri and the Bunurong people of the Kulin Nation, and acknowledge their elders past and present. So thanks again, and Jen has done a fantastic job of starting the introductions, but thanks again to those who are joining us here today in person and online. and it's a great opportunity, I think, to celebrate the many women who have contributed to CIRA's success over many years, and also who have contributed to the reduction in vision loss and blindness in our community. And it's great to be able to share this event with so many special guests and supporters of CIRA. So before we begin, I guess for those of you who are relatively new to our community, I thought I'd explain a little bit about the work that we do here at CIRA. So we are a not-for-profit medical research institute based here in the Eye and Ear Hospital, and we have a joint research strategy with the Eye and Ear Hospital. We're very closely and proudly affiliated with the University of Melbourne, and we rank very highly internationally in the rankings for vision and eye research. And we're also Australia's only World Health Organization collaborating centre for the prevention of blindness, as well as being home to Australia's largest ophthalmic clinical trials centre, and also to the Lions Eye Donation Service, which provides donor tissue for sight-saving corneal transplants and medical research. And our mission is very simple. It's to eliminate the major causes of blindness and to reduce their impact on people's lives. And as I mentioned, our teams are located across the Eye and Ear Hospital here in the main campus, and for just a few more weeks now at the Eye and Ear on the Park, because we have big plans underway, and we're really excited that we're now in the home stretch of a major redevelopment. So we're developing state-of-the-art new labs and a new home for our research nearby at 200 Victoria Parade. And we're also developing a major new clinical trials centre to be known as Cerulean Clinical Trials on level 7, just above us in this hospital, a few floors from where we are today. And this is a sneak peek of our new discovery and research labs, which we hope will become a genetic engineering hub, as well as a hub for other types of research and vision research, and that will be used by our research partners as well as those of us who work on the discovery side of research. So this shows the labs. There's a fantastic view from the 10th floor of that building across the way, which I think will be conducive to good research going on. But I guess the other point is that we need the facilities to be able to attract the next generation of staff to work on the projects that we value, and also to retain the fantastic staff that we already have. So we were very conscious that we'd outgrown the facilities, and we had researchers split around many different sites trying to get their work done. We want to create something which facilitates and enables that work so that we can do work that is really internationally leading with the facilities that we have. So a little bit about our gender action plan. There's no point having all these great facilities if you don't also have the best team and the best people in them. And to ensure that we have a diverse and inclusive workplace culture that includes the opportunities for everyone to deliver the research to the highest quality, part of that process was launching our gender action plan. And I talked about that last year when we actually launched it. And there are a number of things where we're doing pretty well. We have equal representation on our board, on the CIRA board of men and women, and also on our executive team. And of course, we have the amazing achievements of our trailblazers, including Robin Geimer and Penny Allen and Lindell Lim. And we've also added to our senior ranks with more female appointments over the last year. So Michelle Gallagher, who has been appointed as the new head of Cerulean Clinical Trials, is a high-profile tech entrepreneur and digital health expert, well known for her advocacy for women in STEM, and she's been a fantastic appointment. And Michelle is here today, and we're so pleased to have Michelle on board, and I think that's a great area of progress for us in our clinical trials. Also Heather Matchin has been appointed as Director of the Lions Eye Donation Service, and Heather can't be with us today. She's representing us at the annual Corneal Society and Eye Bank meeting. But again, with these two women, we've made senior appointments that strengthen our leadership team within CIRA. And again, as Chief Operating Officer, we have Leah Borsboom and Tina Cheng sharing that role, again providing senior female leadership within our organisation. So I think that's important, and they're providing great leadership to our organisation. But I think we're kidding ourselves, and I said this last year, if we think that there isn't more that needs to be done to achieve gender equity in our organisation. So we know that the medical research funding environment remains very difficult for all of us, but also it's particularly difficult for early career scientists. And within that group, women are disproportionately affected, as are other underrepresented groups. And so, partly to address that, our Equity Fellowship for Excellence in Vision Research is an early step in addressing these challenges. And I'm very excited to be able to hear from Anna shortly about her research plans at CERA as our first Equity Fellow. So thanks again, everyone, for joining us today. And I wish you all the best for International Women's Day. And I hope you enjoy the rest of the morning. And I'll hand back to Jen. Thank you.
Thanks, Keith. CERA's Equity Fellowship has been established with seed funding from CERA's Foundation, which provides philanthropic support to CERA via the many generous donors we have, who leave a gift in their will to vision research. Olivia Hilton is chair of both the CERA board and the CERA Foundation. Unfortunately, she can't be with us today, but wanted to provide this message.
Hello, I'm Olivia Hilton, the chair of the Centre for Eye Research Australia board and also the Foundation board. I'd like to acknowledge the traditional owners of the lands and pay respects to elders past, present and emerging. At CERA, we have a strong commitment to equality and women's issues. This is demonstrated through our equal representation of men and women on our board, strong leadership and representation of women at our executive level, and also some trailblazing women's researchers within our organisation. Despite all of this progress and our commitment, there are still a lot of work to do within our organisation and globally. And that is why the CERA Foundation is investing in the Equity Fellowship. This year, we would like to award that fellowship to Dr. Anna Wang. Congratulations, Anna. This is a demonstration of our continued commitment to this issue and to ensure that women's researchers are given equal opportunities, and we continue to accelerate and commit to this particular issue. Thank you to all of the women at CERA for your incredible commitment, and thank you to everyone that has joined us today. By investing in the Equity Fellowship, we hope to accelerate progress for women.
Thank you, Olivia. As Keith and Olivia have mentioned, CERA is fortunate to benefit from the wisdom and leadership of some trailblazing women, and I'm about to introduce you to one of them. Professor Robin Guymer, AM, is a Deputy Director of CERA and Head of Macular Research. She's also a Professor of Ophthalmology at the University of Melbourne and a Consultant Ophthalmologist at the Eye and Ear Hospital. She's truly the definition of a trailblazer and was recognised as such when she was inducted into the Victorian Honour Roll of Women in 2021. Over the past quarter century, Robin has been part of international research collaborations that have transformed the treatment of age-related macular degeneration. She's consistently ranked in the world's top macular researchers and was recently recognised by the Australian Research magazine as Australia's top researcher in ophthalmology or optometry for 2024. Please join me in welcoming Professor Robin Guymer.
Thanks, Jen, and thank you everyone for attending today. Like the speakers before me, I would like to acknowledge the traditional owners on the land in which we are meeting. Last year, on International Women's Day, I announced plans for CERA's first Equity Fellowship for Excellence in Vision Research. Today, I am delighted to be here to introduce to you our new and first Equity Fellow, Dr. Anna Wang. When I reflect back, a lot has changed since I started research 25 years ago. There's definitely a lot more women since I started, and many more at a senior level. By way of an example, I have an NHMRC synergy grant which aims to bring together multiple disciplines to address a particular research question. And I was just interested in getting the best researchers together, and it just so happens that this research grant is led by 4 senior women. Myself, Erica Fletcher, who coincidentally happened to be Anna's PhD supervisor, and Elise P. Bay from the University of Melbourne, and Melanie Barlow from the Walter and Eliza Hall Institute. Sorry, I'll just see if I can get going. Okay, thank you. To make sure that we were inclusive in this grant, we did have to find some young emerging male researchers to join us. And so we found Zichao Wu and Brendan Ansell to join our team. I'm not sure whether such a research group of 4 senior women at professorial level as chief investigators would have been able to be assembled 25 years ago. So progress is certainly being made, and as many speakers before me have mentioned, there's still more to do. For example, 25 years ago, we were talking about a glass ceiling and getting women into senior roles. But now what's very topical in the media is this new phenomenon called a glass cliff. This is where women are promoted to senior roles at a time of crisis, often making it very difficult for them to succeed. And they're often perceived as the cause of the problem that they've inherited. In our own sector, we seem to have many excellent women study science and medicine, pursue PhDs, but keeping them with us and getting them to reach senior levels is difficult for many reasons, but definitely funding in the mid-career stage is an issue. We end up losing many of them to other careers and industries because we cannot attract the funding that they need in their early stage post-PhD to sustain that research career. And so that brings us to the Equity Fellowship. CIRA's Equity Fellowship for Excellence in Vision Research aims to tackle this problem. The fellowship aims to encourage and develop the career of a researcher with high potential for future leadership in eye research. It's 3 years in duration, provides the continuity and momentum needed to enable competition for externally funded schemes. And it benefits CIRA by ensuring that we can retain an excellent scientist. As Olivia mentioned, Anna joins us from the University of California in Berkeley. I don't want to steal Anna's thunder by talking about her too much, but suffice it to say she's achieved a lot in a very short time. Although she's only a few years postdoctoral, she's already been a first author on a Nature paper. Her research looks at retinal ganglion cells which carry the vision information from the eye to the brain via the optic nerve. She has identified a completely new class of retinal ganglion cells that respond to the direction of movement of objects in our visual world. It's currently completely unknown how these cells are affected by glaucoma, and therefore Anna's work is both groundbreaking and exciting. And it adds a whole new dimension to our Discovery Science program here at CIRA. So please join me now in welcoming and congratulating CIRA's first Equity Fellow, Anna Wang. Thank you.
Thank you so much, Robin, for that lovely introduction, and good morning, everyone. I'm really honoured to be given the chance to talk to you all about me and my research at this important event, just celebrating women. And that is such a lead up. I'm really touched. So what I'm going to talk to you about today is my research into retinal ganglion cell types and their vulnerability in glaucoma. And before launching it into it, I'd like to just acknowledge the Wurundjeri people, who are the traditional custodians of this land. I'd like to pay respects to the elders, both past, present, and emergent, of the Kulin Nation. And I'd like to also highlight the important work being done to spotlight Indigenous women's voices and rights at the Wiyani Yutengani, or Women's Voices, National Summit last year, and the launch of their Change Agenda this year. So, I am, as you have heard, the very happy recipient of the inaugural Equity Fellowship for Excellence in Vision Research. I am incredibly thankful to the CIRA Foundation, whose chair we heard from, Olivia Hilton, and all, of course, the donors who make CIRA's work possible. I'd like to also give many thanks to Robin, Lindell, Keith, and Peter for awarding me the fellowship. This is really an incredible opportunity to do some very exciting and impactful work as an early career researcher, which is really hard. It's a really hard time in a researcher's career. And I really hope to make some important contributions to both CIRA and retinal research in general. So just what I love about being at CIRA is that there are lots of opportunities for collaboration. Many amazing researchers here. We have access to state-of-the-art facilities, and also clearly CIRA is supporting emerging young female scientists such as myself and also other female scientists. And this really gives us the ability to do some high-impact work. And that's great. So to start, I thought I'd just give a background of who I am and where I've been. So I grew up in Sydney, and I did my undergrad at the University of Sydney. And this is where I fell in love with neurons and how they work. I went on to do an honours in neuropharmacology, and you can see this is my honours cohort on our graduation day, which is fun. And this just really sparked a fascination in what's happening in neurons at a cellular level. And after my honours year, I wasn't 100% sure about plunging into a PhD right away. I wanted to get some more experience in a lab. So I spent a year as a research assistant in Singapore, doing some really interesting work looking at the brain. And you can see these are my lab members in these pictures here. And after this, I felt like I had a really strong footing in neuroscience, and I was certain that I wanted to pursue a PhD. So after this, I came back to Melbourne to study the retina with Professor Erica Fletcher. So I've actually always been fascinated by the retina. It has a beautiful structure and circuitry, and most of all, I was really gripped by the possibility of restoring vision. So I started my PhD, and I was looking at retinal cells in aging and disease. And what was apparent to me was that even though we have a lot of studies looking at the retina, it's clear that there's still a lot left to uncover, especially during disease. So after my PhD, I had this great chance to go to UC Berkeley to work as a postdoc with Assistant Professor Teresa Putaseri. In Berkeley, I spent 2.5 years chugging away at a very exciting project using cutting-edge techniques, which I'm very excited to be talking to you about in just a little bit. Thank you. So to go into my research, what I'm now most interested in is getting a better understanding of a retinal disease called glaucoma. This is an example of how vision loss looks or can look like in glaucoma. You can see that it can be very debilitating and severely affect your daily tasks, such as driving. So glaucoma is a disease where cells called retinal ganglion cells progressively die. And I'll come back to that term, retinal ganglion cells. But a major risk factor is increased pressure in the eye. And although we have medications and surgical procedures to lower pressure, this does not prevent the disease progression in many cases. So clearly there's something else involved, and it's important to investigate what other factors are contributing to ganglion cell death. There is an estimated 69 million people affected worldwide, and what I think is very striking is that this rate hasn't reduced in the last decade. And there's $4.3 billion estimated cost in Australia alone by this disease by 2025. So I think there's real incentive to look further into this disease. So let's focus on the retina, which is a thin layer of neural tissue found at the back of the eye. Zooming in, we can see that the retina is made up of several different cell types arranged in layers. It's a bit like a layer cake. The photoreceptors are at the top there in purple, and they convert light energy into electrical signals which pass through and reach the retinal ganglion cells, seen here in yellow. So they're at the bottom. So retinal ganglion cells are responsible for sending the information from the retina to the brain, as Robin mentioned earlier. So they do this by sending the signal along these long projections called axons that make up the optic nerve. The optic nerve then travels to visual centers in the brain. But these retinal ganglion cells, they're not just passively transmitting this signal. We actually have a much greater understanding of their very role. So we know that there are many different types of retinal ganglion cells. So in mice, we know that there are over 30 different types, and recently studies have estimated up to 18 types in humans and non-human primates. So why do we have these different types? Well, actually, these different types of ganglion cells are responsible for processing different aspects of vision. So what's that mean? Well, let's take this image here, which is a still from the movie Darjeeling Limited, where Adrian Brody is running for the train. So we have some ganglion cells that will transmit information about the contrast of the image. Some will transmit color information, and it gets more complex. Some will transmit information about edges. In mice, there are retinal ganglion cell types that respond to a looming visual stimulus, which is important for them for detecting a predator that might appear above them. So you can see that these cells are really much more than just a conduit. They're doing some really intensive processing here. So we know that retinal ganglion cells are damaged in glaucoma. There is increasing evidence from mouse studies that some ganglion cell types may be more vulnerable than others in these models of glaucoma. And what I think is very exciting is that this is something that looks like it might also be happening in patients. And this spectrum of vulnerability in retinal ganglion cells is something that I'm very interested in exploring in my research. So we know that we have up to 18 different types of retinal ganglion cells according to current estimates. But we actually only have a really good understanding of around 4 of these types. That's less than a quarter of the total. So you can see there's a big hurdle to understanding glaucoma, and that is first getting an understanding or lay of the land and characterizing some of these unknown cell types. And actually, so what I was doing in Berkeley, my previous work, was exactly that, characterizing a retinal ganglion cell type that had not previously been seen before in primate retina. So these are called an on-direction selective ganglion cell, and these cells are essential for gaze stabilization. So gaze stabilization is important for keeping the image of the world stable as it moves around you. And the classic example is looking out the car window or train window at trees or telephone poles, and your eyes will make this rapid to and fro movement like we see here. Without gaze stabilization, our eyes would have repetitive, uncontrolled movements and result in blurry vision, dizziness, loss of balance. So we do know that these cells have been described in mouse retina, but for a long time they were thought not to exist in primate retina. They thought that all this processing was being done by the brain. So it was a very exciting finding that we were able to find this cell in the retina, and our data was actually also very strong. And this allowed us to publish this exciting result in Nature. So, okay, great. We got a new ganglion cell type. What's this mean? So we now also understand a whole new level of processing that is happening in the retina, where we are processing this gaze stabilization. It begins there. We can be better informed about strategies for vision restoration. So to better know what we need to restore, we need to know what's there. This has implications for eye disease, for example, infantile nystagmus, which is the constant involuntary to and fro movement of the eyes, and also in glaucoma, like Robert mentioned before. We don't know what's happening with these cells in glaucoma. Finally, I'd like to bring the technique that we used in Berkeley to help identify and characterize these cells to SIRA. I think it's a very powerful technique that can tell us a lot about these ganglion cell types. So I'd just tell you a little bit about what we did was, so we dissected the retina and we kept these cells alive in an oxygenated solution that provides the retina with the perfect mix of nutrients. And so in this way, even though the cells are out of the eye, they are still functioning as they normally would. We can then image these cells using a very powerful microscope, which is able to use an infrared laser to capture the function of these cells. So this is an example of what sort of videos we get from this imaging. Each of these round shapes is a cell. And you can see a lot of these cells lighting up when the light goes on, and there are some that go off instead. So this pair of cells you can sort of see working in opposition when the light goes on. So we can identify different patterns of function in this way But we still need to work out which ganglion cell types they correspond to. So what we can do is then take this tissue and add combinations of fluorescent dyes that are drawn to particular cell types. And in this way, we can light up those dyes and see what cell types we have. So we can apply a variety, and looking at the pattern of expression of these markers, we can really pinpoint what ganglion cell type we have. And that way, we can match up functionality to type. And this is really important in this characterization of ganglion cell types that we need to do. So we know that there are different ganglion cell types in glaucoma, and different ganglion cell types show different vulnerability to the stresses of glaucoma, and that can lead to their death. This is, I think, a very interesting aspect of this research to explore further, because if we can identify which cells are most vulnerable, we can target early diagnostic testing towards the functions of those cells. Furthermore, I'm very interested in also investigating why we're seeing this different vulnerability, and in that way work on alternative treatments. So the progress that I have so far is to optimize the molecular markers used for the identification of ganglion cell types. And what I hope to bring to CIRA is a setup much like what we had in Berkeley. This is a photo of our lab there, and we have a microscope on the left of the screen where we put this retina. And then on the right, we have a little screen that we can project a visual stimulus onto the retina. So this, of course, is great for what I'm interested in, and it can also open up treatment targets and also, open up working with human donor tissue. And I can see, this having a utilization for many other researchers here at CIRA as well. In terms of that, I'm so very excited to be connecting with world experts at CIRA who are also studying how ganglion cells degenerate in glaucoma and how they can be restored. I think collaboration is really key in today's research landscape as we come to examine diseases from many different angles. And finally, I'd like to acknowledge some of the amazing female mentors I've had in my career so far. So from the University of Melbourne, Professor Erica Fletcher, Dr. Kirsten Vesey, and Dr. Patricia Youssef guided me throughout my PhD. They were really instrumental for me learning the ropes, learning all about the retina, the lab work, the writing, the presenting, and also providing the moral support that you need during a PhD. Also, from the University of California, Berkeley, Assistant Professor Teresa Putaseri. Her advice and support during my postdoc was critical, and her enthusiasm for science was infectious. So these mentors have really shaped who I am as a scientist, and I really hope to pass on that guidance and enthusiasm to the next generation of young researchers. So thank you very much for listening to my talk. That concludes the presentation, and I'll take any questions from the room or online. We have Callum in the auditorium with the microphone, and Richard moderating questions online, so please raise your hand, and yeah.
Thank you, Anna. That was really inspiring to all our younger PhD students here. I'm just wondering, that cool setup you showed us with the microscope.
Yep.
Do you want to build something similar to that here at CIRA? And how long do you think that will take?
Great question. I see a collaboration brewing. So we actually have access to the two-photon microscope at the Peter Doherty Institute, and what we would need to add on are most of this stuff is very simple to acquire. And so I imagine that setting this up and making sure that it is running smoothly would maybe take a year or so, and then, then the world's your oyster.
Sorry, I just had to make Callum run as far as possible with the microphone. That was a wonderful talk. Thank you so much. I was really interested in what you were mentioning about finding new therapeutic targets. So is the idea that if we can characterise these different retinal ganglion cells, that you might be able to, for example, have a drug which could specifically target that type of cell?
Yeah, I think that is one of the hopes of what this research could bring. Because we know that there are different types, they might express different proteins that make that type unique but also targetable. So yeah, exactly. I think that would be fantastic.
Hi Hannah, lovely to hear your work and great presentation.
Thank you.
I'm also interested in the cells like feeding into the ganglion cells, which
Yes.
I know you haven't talked about here, but how are you going to approach, or do you have any plans to approach, all the different combinations that feed into your ganglion cells, so your bipolar and amacrine and so forth, and how much do you need to look backwards up the pathway?
Yeah, that's a fantastic question. Because exactly as you mentioned, retinal ganglion cells are sort of pooling all this output from these other cells. And we know that maybe amacrine cells are also potentially vulnerable in glaucoma and diabetic retinopathy, other retinal eye diseases. So I think there are definitely ways to look at that with the setup that we have, but sort of exposing all of those other cells to be visible. Because I think that is the main sort of hurdle with studying those other cells in this way. How do you see them? And so there's definitely ways that we can make those cells visible.
I think we have a question in the front row.
Yeah.
Congratulations, Anna. It's so wonderful to see such an enthusiastic, wonderful young scientist. Congratulations. My question is about eye pressure. I'm not sure if I heard you correctly, but you mentioned that eye pressure can cause a problem with the glaucoma? Yes. So what does this mean? I haven't heard of this before.
Yeah, right. So in glaucoma, an increased intraocular pressure is a very common risk factor and in glaucoma. And so what that means is that if there's increased pressure in the eye, the sort of anatomical structure can cause damage to the retinal ganglion cells. So all of their axons, which I mentioned earlier, is is the optic nerve, essentially. The optic nerve can sort of be squished, I suppose, by this intraocular pressure. And in that way, you can damage the ganglion cells. I hope that's...
And what can cause pressure? Is it something that's just there, or different factors can cause it?
Yes, that's a great question. You have primary and secondary glaucoma, so we don't really know in terms of primary glaucoma. Otherwise, it might be something like surgery or other factors as well. So, yeah.
It's Women's Day. Thanks, Anna. It was a very nice presentation. And just thinking, the implications will go wider than glaucoma, I think, because in AMD we're trying to hijack the ganglion cells to make them work as photoreceptors. And it may well be that you have a particular sort of your 18 that might be a better one to target, for example, more robust. As opposed to one of the others. So I think I can see your work trickling down to other areas that we're interested in, Sarah.
Absolutely, exactly. It's a great point.
And Anna, great work and very nicely presented. And when it comes to pressure in glaucoma, we're pretty good at lowering pressure in glaucoma. And so it's great to see that your work is focusing on other aspects of the condition, because we know that for many of our patients, even when we do lower the pressure in the eye, they still continue to get worse. Exactly. And so that's why it's so important to understand why that might be happening. And your work gives us a different insight into that, which I think is fantastic.
Yeah, thank you. Is this the last question? Yes? Oh, last one. Okay.
Congratulations, Anna.
Thank you.
It's a pleasure that you're here with us. We are very happy for having you. Sometimes when we are working in basic science to understand things, we have the limitation to look at the human retinas, right? And the human eyes.
Yes.
So I understood that with your technique, it's going to let us to give a step forward, right?
Yes.
Because we will be able, I would like that you comment on it.
Yes. So it's very difficult to look at, these retinal ganglion cells in human tissue. But we are so lucky at CIRA to be partnered with the Lion's Eye Donation Service, and we can have a look at human donor tissue using this technique, which is incredible because this is something that is so difficult and very, very, very few labs anywhere in the world are able to do this. So that's a great question, and I think that would be very exciting for the future. Okay, great. Thank you very much. I'm happy to answer any questions via email or at morning tea. So thank you.
Congratulations, Anna, on a fantastic presentation. I'd now like to invite co-chairs of CIRA's Gender, Equity, Diversity and Inclusion Committee, Professors Peter van Wijngaarden and Lindell Lim, to come up to present a small token of CIRA's appreciation to Anna.
Come on up, Anna.
Come on back.
Congrats.
Congratulations. Thank you.
Congrats. Great. So I'm sure that you all enjoyed Anna's presentation as much as I did. So I'd like to congratulate you again, Anna. So as one of the co-chairs of the Gender, Equity, Diversity and Inclusion Committee, I'm really thrilled that this fellowship marks a first and very meaningful step for our committee and our commitment to gender equity. We are working on and embarking on a gender action plan, which we see as an ongoing plan for CIRA. Striving for equity as part of that action plan is not just an issue for women, nor is it some sort of HR exercise to tick a box. It's something that's really core to our strategic mission at CIRA, and it's something that each and every one of us is really pleased to be part of. We've got really passionate people in our Gender Equity Committee, and it's just a privilege for Lindell and I to play a small part in leading that initiative. Our Gender Action Plan is really strongly focused on attracting and retaining the best women researchers and professional staff. We often get to highlight and see the achievements of our research staff, but we have a legion of unsung heroes in our professional staff behind the scenes, without whom we just could not function as a research organisation. So I'd like to acknowledge them. But beyond attracting and retaining the very best women to our organisation, we really need to enable them to advance their careers, tackling those entrenched problems that women face with inequities in promotion and leadership. So we're really committed to that, and I think we've heard about it, but it's worth reflecting on that CIRA really does a great job of that. I have the privilege of working in a really dynamic executive, which is enriched so very much by having equal representation of men and women, and that's just a joy and something that keeps me motivated and excited about being here. So equally, our plan's about delivering great science that benefits women, and that's something that is engineered from the level of the culture of the organisation, but also in how we design our research and conduct our research. So it's now my pleasure to hand the podium over to my fellow co-chair, Lindell Lim, who's going to tell us about a project that has the eye health of women right at its heart. So welcome, Lindell. Thank you.
Thank you very much, Peter. Like my previous speakers, I also like to acknowledge the traditional owners of the land and pay my respects to their elders past, present and emerging. And I'm really excited here today to actually talk to you about a new project that is aimed to improve the eye health of women living with diabetes who become pregnant. And this has only become possible due to the generosity of Jackie Marie and George Stamos AM from GJK Facility Services, without which we would not be able to do this project. And this project has actually stemmed from a very long-standing interest of mine with regards to finding out what pregnancy does to a number of different eye diseases. And this actually started a long time ago when I was pregnant at the same time as one of my other patients. And whilst we were standing there complaining about our sore backs, how big our feet were, how sore our bellies were, she actually said, "Doc, pregnancy's doing all of this to our bodies. What's it going to do to my eye disease?" And she had quite a rare eye disease. And I actually sort of said, "You know what? I'm not sure, but I'm going to find out." And I did after many years. But extending from that is this new project. And it's actually built upon the work of my PhD student who actually published recently about how common diabetic retinopathy is in pregnant women with diabetes and how it progresses during that time. And during that work, we published that last year, and during that work we actually found that less than 50% of the women who were pregnant with diabetes were able to actually get their eyes checked at the frequency that they needed. And this was because of all of the other competing needs and medical appointments that they actually had during that time. They had to see, obviously, their obstetrician, their GP, their endocrinologist, their diabetic educator, their nurse, their podiatrist, and their ophthalmologist. It was really, really difficult. As one of them actually said, it's like having a full-time job on top of my full-time job. And so we thought, okay, myself and my colleague, Associate Professor Andrew Simons here, we thought we can do this better. And we decided that what we need to do is actually bring the eye screening into the one doctor they would see more often, which was their obstetrician. And so that's what we're doing. So we've got here a brand new AI-assisted retinal camera, which was purchased with the generosity of both George and Jackie Marie. Thank you very much. And I'd like to invite, actually, George and Jackie Marie up to the podium here so we can actually launch our project with Associate Professor Andrew Simons. Thank you very much.
You're always like. Sound damage. Yeah.
Thank you, Jackie Marie and George, for your incredible generosity. It will make a huge difference. Thank you to everybody who's here with us in person and online today to celebrate International Women's Day. I'd like to conclude today's proceedings with a favourite quote of mine from feminist writer Susan Sontag. Women have another choice. They can aspire to be wise, not merely nice, to be competent, not merely helpful, to be strong, not merely graceful. They can be ambitious for themselves, not merely for themselves in relation to men and children. This was written many decades ago, but for me, the sentiment resonates today. In closing today's event, I pay tribute to wise, competent and strong women everywhere. Our ambition for ourselves can accelerate progress and make a huge difference in vision research, in science and beyond. Thank you. That concludes the formal proceedings of our event, but please stay and join us for morning tea outside.